Distribution of single nucleotide polymorphism. Genetic variations in DNA sequences when there is a variation at a single nucleotide. So if there is a variation at a single point, then it is called as single nucleotide polymorphism. In this diagram, we can see that what is single nucleotide polymorphism. Here we can see that there are different versions of the DNA. These DNA, they belong to different individuals. So this is the first version, second version, third and fourth. We can see in the first individual at a particular point there is adenine, while in the second there is cytosine, while in the third there is guanine, while in the fourth there is thymine. So remaining all the sequences, they are similar in all the four individuals or in four versions of the DNA, but there is a variation at a single point. If there is a variation at a single point of the DNA, then it is called as single nucleotide polymorphism. This is the percentage of distribution of different nucleotides in the DNA on the genome. In case of guanine, that is 25%, distribution is 25% or variation. In case of cytosine, it is 34%. In case of adenine, it is 40%, while in case of thymine, it is just 1%. So this is the percentage distribution of all the nucleotides which are present in our genome. These are the number of SNPs which are present on the human genome. If we see that there are approximately 25,000 SNPs which are non-synonymous, while there are 50,000 SNPs which are synonymous. There are 25,000 SNPs which are present in the regulatory regions. Regulatory regions are those regions which are adjacent to the genes, from where gene is regulated. So those regions are called as regulatory regions. 50,000 are the SNPs which are intragenic. Intragenic SNPs are those SNPs which are present on the DNA but between two genes. If the SNP is present between two genes, then it is called as intragenic. While there are 50,000 SNPs which are distributed in intergenic. Intergenic SNPs are those SNPs which are present in the coding sequence of the DNA. So if we combine all this information, we can say that there are approximately 25,000 SNPs which are non-synonymous, 50,000 they are synonymous, 25,000 are present in the regulatory region, 50,000 are present intragenic between two genes, and there may be approximately 50,000 SNPs which are present within the genes. Human SNP distribution, most common changes, they are in the form of transitions or in the form of transversions. And second type is single nucleotide insertions and deletions. So transitions are those changes where purine are converted to purine. If purine is converted to purine and pyrimidine is converted to pyrimidine, then such a change of the nucleotide or change of the base is called as transitions. While on the other hand, if purines are converted to pyrimidines and vice versa, pyrimidines are converted to purines, then this kind of the change is called as transversions. So in the sequence of the DNA, transitions and transversions, both type of the changes are present. Along with transitions and transversions, there are single base insertions and deletions, which are also called as indels. How SNPs are indicated? So there is a code or pattern that has been designed that how a nucleotide will be represented, or if there is a change, if there is one base or the alternative base, how to mention that change. So A is represented by A, C by C, G by G, T by T. So if there is a variation between A and C, it will be represented by M. A and G, if there is any one of the nucleotide is replaced by another, it will be represented by R. If there is A or T, it will be represented by W, and so on. So this is a pattern that how if there is a change, one nucleotide is replaced by another nucleotide, so how we have to mention that change. Like again take the example of A and C. If there is A and C, if there is A or if there is C, so we can write in that place by writing the English letter M. So M is represented either by A or by C. There are different SNP related databases which has been designed. Some of the most common databases, they are for ataxia telangiectasia, related to the breast cancer, related to the cystic fibrosis, related to the cyto. So these are all databases where all the information or all the SNPs which have been identified in particular diseases are present on these databases. So some other databases related to some other diseases have been mentioned here. So we can conclude that SNPs are found throughout the genome, and usually SNP is a single nucleotide change on the genomes.